my screen, right? Okay. So where is the share? Let me find it. Share screen. And also my presentation. And share. So can you see my screen now?
Yes.
Okay, great. And then I will put it on the presentation mode. So do you see it fine? Like, is it in the presentation mode?
Yeah, but we can see your next slide and the notes, Jenny.
You can see the notes or not?
Yeah, I can see
Okay.
the notes and your next slide. I think you need to.
Okay. Maybe let me swap.
Yeah. Okay.
And the next slide. Yeah, it seems strange. I mean, let me do it. So the next slide, can you see my notes or not?
No, it's okay now.
So it's okay now. Okay. Maybe let me swap my monitor because I have two monitors. Okay. Let me stop now. And try it again. Stop share. And I needed to change my display. But it's kind of strange because for my other presentations, it's fine. Maybe I don't switch. I just do this way.
Okay.
So rearrange, select. So maybe let me try it again and to see. So share my screen. Always block my view. Yeah. And then display mode. So how about now? Do you see my notes or it's the presentation mode?
No, we see or not, Jenny?
What?
We see or not? We can see or not?
So I swap it, it will not, right? Oh,
yeah.
Okay. Yeah, I think that's fine. Maybe I will just do this way. So, and it's, how about the next slide?
It's fine now. We don't see any notes.
So it's good. So it's the presentation mode. Okay. So I will stop sharing. Okay. Thanks a lot.
Thank you.
Hello, good morning, everyone.
Good morning, Bu Dita.
How are you?
Good, Ibu. Bu Dita, I would like to introduce you to our speaker. It's Dr. Jenny from USP. There's also Dr. Karen from TGA and Dr. Christian from USP.
Okay, thank you very much for joining our event today. I think it's very important to us. And thank you for all support.
Welcome.
A pleasure.
Yeah. Yeah.
Just a moment, I will change my background first. Ibu Ella, Ibu Dirjen will open this meeting. I think we have to wait maybe 5 minutes. I will contact her first because now she's in US. Please wait for a minute, please.
Okay, Bu. Thank you.
Oke, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. May peace be upon us. Excellencies, distinguished participants, ladies and gentlemen, allow me to greet the dignitaries here with us through the Zoom meeting. Your Excellency, Director General of Pharmaceutical and Medical Devices, Ibu Dr. Anda Lucia Rizka Andalusia,
Suaranya kurang
Farmers.
terdengar, Okti.
Izin Pak, apakah sudah lebih terdengar? Sudah terdengar. Oke, baik. Izin saya lanjutkan. Excellency, Director for Pharmaceutical Production and Distribution, Ibu Dita Novianti Sugandi Argadireja, SSIA PTMM, Associate Director for Government Policy and Regulatory Affairs of Global External Affairs, IFEC, the United States Pharmacopoeia, Mrs. Ruth Lee, Manager of Strategic Customer Development from USP Indonesia, Mrs. Ella Gondowijaya.
Suaranya tidak terdengar, Okti.
Baik, mohon izin sebentar. Baik, mohon izin, apakah lebih terdengar suara saya?
Ya, lebih baik.
Oke. Oke, baik. Mohon izin akan saya ulangi untuk sapaan. Assalamualaikum warahmatullahi wabarakatuh. Excellencies, distinguished participants, ladies and gentlemen, allow me to greet the dignitaries here with us through the Zoom meeting. Your Excellency, Director General of Pharmaceutical and Medical Devices, Ibu Dr. Anda Lucia Rizka Andalusia, PTM Farmers, Excellency, Director for Pharmaceutical Production and Distribution, Ibu Dita Novianti Sugandi Argadireja, SSI APTMM, Associate Director for Government Policy and Regulatory Affairs of Global External Affairs, IFEC, the United States Pharmacopoeia, Mrs. Ruth Lee, Manager of Strategic Customer Development from USP Indonesia, Mrs. Ella Gondowijaya, our distinguished speakers, distinguished senior officials of the Indonesian Ministry of Health and Indonesian FDA, head of association and organization or representatives, practitioners and academics, and ladies and gentlemen. We would like to extend our gratitude and appreciation for the presence in this wonderful occasion. Welcome to the workshop on ethylene glycol, diethylene glycol, and elemental impurities testing. We are honored to welcome you all at this online session through Zoom meeting today, 25th of September, 2024. First and foremost, let us praise and thank God for His mercy and grace so we can gather here today. Today's event is hosted and organized by Directorate General of Pharmaceutical and Medical Devices, Ministry of Health of Republic of Indonesia, in collaboration with the United States Pharmacopoeia and Australia Therapeutic Goods Administration. The objective of this workshop is to share an overview of the USP General Chapter for EG-DEG testing and elemental impurities testing, and the importance of performing this testing in regulatory perspective. Through this great opportunity, it is expected that Indonesian stakeholders could learn from international experts about the EG-DEG testing and elemental impurities testing on current regulatory. I am Okti Aliviana. It is with great honor I can be your MC for today's event. We have an exciting panel of speakers for you today, and we hope that you will find their presentation and discussions informative and engaging. Before starting the session, let us pray together for the success of this event. Pray based on individual belief. Please begin. Thank you. Excellencies, distinguished participants, ladies and gentlemen, now we will have a remark for this important occasion from Excellency, Director General of Pharmaceutical and Medical Devices, that will also open this workshop officially. Please welcome Ibu Dr. Anda Lucia Rizka Andalusia, APT Empor Mars. Okay, please wait a moment. Welcome Ibu Dirjen.
Iya, iya. Maaf, saya sempat terputus ya tadi. Oke. Kita bisa mulai. Tadi saya sempat terputus. Halo.
Ya, bisa Ibu. Silakan Ibu.
Silakan langsung remark, Ibu.
Oke. Ya. Oke, good afternoon, good morning everyone. Honorable guest, Senior Manager Scientific Affairs, EMEA, the United States Pharmacopoeia, Dr. Christian Zeine, Principal Scientist Science Excipients, USA, the United States Pharmacopoeia, Dr. Jenny Liu, Assistant Director Senior Regulatory Scientist Quality Australia, TGA, Dr. Karen Love, Associate Director Governor Policy and Regulatory Affairs, Global External Affairs, APEC, the United States Pharmacopoeia, Ruth Lee Chu AI, Manager Strategic Customer Development USP Indonesia, Ibu Ella Gondawijaya, representative from Indonesia, FDA, representative from Faculty of Pharmacy across Indonesia, team expert of Pharmacopoeia Indonesia, representative from Association Pharmaceutical Industry, and testing laboratory in Indonesia. Very good morning. Assalamualaikum warahmatullahi wabarakatuh. First, praise to God Almighty for His grace and blessing, who has granted us the opportunity to hold workshop on ethylene glycol and diethylene glycol, or EG-DEG, and elemental impurities testing, which is held through collaboration between the Indonesian Ministry of Health, the United States Pharmacopoeia, and the Therapeutic Goods Administration, TGA Australia. We greatly value the support and facilitation provided by USP and TGA Australia in organizing this workshop. Their commitment and efforts have given us the opportunity to deepen our understanding of critical issues concerning the testing of ethylene glycol and diethylene glycol and elemental impurities, which are vital to ensuring the quality and safety of pharmaceutical products. Ladies and gentlemen, esteemed guests, first and foremost, I would like to extend my heartfelt thanks to all the distinguished guests, speakers, and participants who have taken the time to join us today. Your presence is a great honor, and we are grateful for your dedication to advancing the knowledge and understanding in ethylene glycol and diethylene glycol and elemental impurities testing. Ladies and gentlemen, esteemed guests, medicine are a crucial component in supporting the national healthcare system. As mandated by Health Law Number 17, year 2023, medicine must meet the standard of safety, efficacy, and quality, and also availability at affordable prices, along with the guarantee of halal product. It is important to remember that this assurance relies not only on regulation, but also on the integrity of the production and testing processes conducted by all stakeholders. The case of atypical progressive acute kidney injury in Indonesia serves as a stark reminder of the importance of supervision and quality assurance. Excessive contamination of ethylene glycol and diethylene glycol has been proven to be the main cause of acute kidney injury cases, which have resulted in death and suffering for many families. In response, the Ministry of Health has mandated testing for EG and DEG contamination, both in raw material and finished product, as a preventive measure. The second supplement of the Indonesian Pharmacopoeia edition, 6th edition, has also been updated to ensure this provision are implemented. Ladies and gentlemen, esteemed guests, in this modern era, the development of science and technology continues to advance rapidly. The types and form of medicine distributed in Indonesia are becoming more diverse. So the quality and testing standard used in the Indonesian Pharmacopoeia 6th edition and its supplement must be continuously updated and aligned with international standard. The Ministry of Health published the third supplement of the Indonesian 6th Indonesian Pharmacopoeia, 6th edition Indonesian Pharmacopoeia in 2024, which includes change in monograph, analysis methods, and regulation expected to facilitate the pharmaceutical industry in ensuring that the medicine they produce are safe, high quality, and efficacious. Furthermore, this third supplement has also added methods for testing elemental impurities. This method is becoming increasingly important amid global demand for safer and higher quality pharmaceutical products. With this workshop, I hope that we can all enhance our understanding and ability to apply impurities testing and standard food for EG and DEG and elemental impurities, thus providing stronger assurance of safe and beneficial medicine for Indonesian people. Ladies and gentlemen, esteemed guests, this workshop is form of our commitment to enhancing our understanding of the urgency and implementation of EG and DEG and elemental impurities testing, guided by international experts to ensure the public receive quality assured medicine. I would like to express my highest appreciation to all parties who have contributed to organize this event. May all our efforts to make Indonesia healthier and stronger always receive blessing for the God Almighty. I hereby declare the workshop on ethylene glycol, diethylene glycol, and elemental impurities testing is open. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you, Ibu Dirjen, for your profound speech. Excellencies, distinguished participants, ladies and gentlemen, to continue our agenda today, we will move to the panel session. To guide this session, I respectfully invite Leader for Price Control and Drug Market Monitoring Working Group. Please welcome Ibu Elza Gustanti, SH SSI APTMH, as moderator for this session.
Thank you, Opti. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Allow me to greet the dignitaries here with us through the Zoom meeting. Your Excellency, Director General of Pharmaceutical and Medical Devices, Ibu Dr. Randa L. Rizka Andalusia, Apoteker M.Pharm Mars, Excellency Director for Pharmaceutical Production and Distribution, Ibu Dita Novianti Sugandi, Argadireja, SSI APTMM, Associate Director for Government Policy and Regulatory Affairs of Global External Affairs Asia Pacific, the United States Pharmacopoeia, Mrs. Ruth Lee, Manager of Strategic Customer Development, USP Indonesia, Mrs. Ella Gondo Wijaya, the Honorable our speakers, Senior Manager Scientific Affairs, EMEA, the United States Pharmacopoeia, Dr. Christian Zane, Principal Scientist Science, excipient USA, the United States Pharmacopoeia, Dr. Jenny Liu, Assistant Director Senior Regulatory Scientist, Quality, Australia Therapeutic Goods Administration, TGA, Dr. Karen Love, Excellency, distinguished all participants who joined in this workshop. Representative from FDA Indonesia, Balai POM, Lokapom, pharmaceutical industry, Faculty of Pharmacy, and lab testing laboratory. Ladies and gentlemen, welcome to our online workshop on ethylene glycol, diethylene glycol, EG-DEG, and elemental impurities testing, which is held through collaboration between Ministry of Health of Republic Indonesia, the United States Pharmacopoeia USP, and Therapeutic Goods Administration TGA Australia. I am Elza Gustanti, and it is a great honor for me to be your moderator today. We are excited to have a wonderful lineup of speakers who will share insight on ethylene glycol, diethylene glycol, EG-DEG, and elemental impurities testing. Ladies and gentlemen, for our session today, we will 3 sessions. Each session there is 1 speaker will be presentation for 30 minutes and then follow discussion for 10 minutes. Okay, we will be start with presentation from Dr. Jenny Liu from USP USA, who will be presenting about EG-DEG testing in excipient, followed by Q&A session. For the second speaker, we will have Dr. Karen Love from Australia TGA, who will be presenting about regulatory perspective on quality assessment of drug product with respect to EG-DEG as well as elemental impurities, followed by Q&A session. And for the last speaker, we will hear from Dr. Christian Zen from USP Europe, who will be presenting about elemental impurities testing and also followed by Q&A session. Ladies and gentlemen, now without further ado, I would like to introduce our first speaker. Yeah, here we have Dr. Jenny Liu from USP USA. Dr. Jenny Liu holds a PhD in chemistry from Boston University and has more than 20 years of experience in analytical method development and validation for pharmaceutical products. She joined in the United States Pharmacopoeia USP in 2013 and is currently a senior principal scientist in the excipient department science division. She is a documentary standard scientist responsible for the development, modernization, and harmonization of excipient monographs and excipient-related general chapter under the work plan of Pharmaceutical Discussion Group PDG, which strive for harmonization among the European Pharmacopoeia, the Indian Pharmacopoeia, the Japanese Pharmacopoeia, and the United States Pharmacopoeia. Yeah, Dr. Jenny Liu will be presenting about EG-DEG testing in excipient. Please welcome Dr. Jenny Liu. You have 30 minutes to present your presentation, please.
Yes, thank you very much, Elsa. I will share my screen now. So I will put it on the display mode. So please let me know if it's working. So is it the presentation mode on your side, Elsa? Is it right?
You need to.
Can you see my
can see-
screen?
You can see the note.
Or do I need to swap my
Yes.
monitor?
Yes, yes.
Okay. Oh, sorry. Sorry, I think I made it wrong. Apologize for that. Share and display mode. Yeah, actually, I needed to share. Okay. then 13. So yeah. So can you see it on the display mode?
Yeah. Yes, Dr. Jenny.
Okay. Okay. Yeah. So it is my pleasure to talk about the yes. Why it is like this way? So it is my pleasure to talk about the importance of excipient quality, USP's perspective on DEG and EG testing of high-risk excipients. In this presentation, I will talk about what is USP's mission and the role, and what's the importance of excipient quality, and also why it is so urgent for WHO to issue alerts and FDA issue guidance on diethylene glycol and ethylene glycol testing of high-risk excipients. And the third, what are USP's perspective and activities? And also I will talk about the pharmacopoeia discussion group harmonization in terms of glycerin, propylene glycol, polyethylene glycol, EG-DEG testing method, and the next steps and the summary. So let's begin. So USP was founded by 11 physicians back to 1820. USP's enduring mission is to improve global health through public standards and related programs that help ensure the quality, safety, and benefit of medicines and food. So since then, USP has been setting public standards for more than 200 years. So USP standards are enforced by US FDA, and they are internationally recognized and globally focused. The USP is headquartered in Rockville, Maryland, near Washington, D.C., NIH, and FDA. And we also have laboratory facilities and offices in US, India, Switzerland, China, Brazil, and Singapore. There are more than 1,300 employees worldwide, and the USP has been working with more than 1,000 volunteers, including scientists, practitioners, regulators, to I think somehow it blocks my view for the public standard setting. So USP actually is declared in the US law according to the US FD&C Act 501B and 502E3B, adulterated drugs and devices. So a drug with a name recognized in the US PNF must comply with the compendial identity or be deemed adulterated, misbranded, or both. So it cannot label away from identity. Identity is very important. Also, the standards must comply with the compendial standards for strength, quality, and purity, unless it is labeled to show all differences according to the 50B and 21 CFR 299.5. So USP FDA and the C Act also indicate to avoid being deemed misbranded, drugs recognized in USP&F must also be packaged and labeled in compliance with compendial standards. Please note, USP standards are enforced by US FDA and other government authorities in the US or elsewhere. USP has no role in enforcement. Why excipients are so important? Because excipients can make up to about 90% of the total mass or volume of medicinal products. Excipients also have many functions as shown in the USP&F functional categories. So they can be used as coating agents, diluent, solvent, flavors, etc. And excipients can help ensure that API is delivered to the site of action. However, complex, non-transparent supply chains can lead to economically motivated or adulterated contamination and adulteration. So supply chain is very important. So USP has been collaborating with stakeholders, including the FDA, to work on the excipient quality. We have received several letters from the US FDA to update excipient monographs and chapters because some monographs lack of specificity of identification tests, and some excipients are well characterized in traditional applications in terms of chemistry. However, they need additional characterization in some of the newer applications, for example, long-acting injectables, vaccines, advanced biologics, and therapeutics, etc. Now let's talk about WHO alerts and FDA guidance for diethylene glycol and ethylene glycol testing of high-risk excipients, especially YID test. In fact, in May 2023, FDA issued a guidance which is testing of glycerol and propylene glycol maltitol solution, hydrogenated starch hydrolysate sorbitol solution, and other high-risk drug components for diethylene glycol and ethylene glycol, and it is for immediate implementation. So why it is so urgent? So in fact, in the beginning of 2023, WHO urged, had an alert to urge action to protect children from contaminated medicines because diethylene glycol and poisoning issues has occurred in several countries and caused more than 300 fatalities in 3 of these countries. So it is so sad, but this issue has occurred a long time ago. Back to 1937, 107 patients died due to sulfonamide elixir poisoning due to DEG adulteration. And to prevent this disaster happen again, US Congress established the modern FDA and C Act in 1938, and also confirmed the USP's official status in the law. So USP has been setting law-enforced public standards. So after that, many incidents occurred in different countries and in different years. Especially recently, the incidents happen again. And you can see even in this year, in 2024, WHO issued another alert about the falsified propylene glycol found in Pakistan, and also cough syrups were recalled due to poisoning, due to the DEG and EG contamination in Africa countries. So these are the resources for these incidents. So USP actually received several letters back to 2007 and 2009 for the similar issue regarding glycerin and propylene glycol, etc. And in this year, USP also received a letter from FDA regarding the update of monographs for polyethylene glycol and polyethylene glycol 40 castor oil. And in these letters, FDA requests USP to implement the EG-DEG test in the identification section. So why ID test? Because this is the impurity test. Why the US FDA requests USP to implement it in the ID section? So because if EG-DEG detection and the quantification is part of the ID section, according to CGMP regulations at 21 CFR 211.84D1, the manufacturers and the drug products manufacturers need to detect and quantify any EG-DEG for each lot received for high-risk excipients, for example, glycerin, propylene glycol, or polyethylene glycol, etc. On the other hand, if the EG-DEG detection and quantification is only part of the impurity test, manufacturers need not include as part of its identity testing the detection and the quantification of EG-DEG for each batch of high-risk drug components. In addition, a manufacturer could deviate from the impurity requirements established in the monograph by labeling the product to indicate that it deviates from the USP test requirements. That's not what FDA wants. They require to test each lot for each container of EG-DEG testing for these high-risk excipients. So we need to understand an important concept, that is excipients are also drug components. In FDA nomenclature, excipient is also known as an inactive ingredient. However, more importantly, FDA considers both active ingredients and excipients to be drug components. According to 21 CFR 210.3, component means any ingredient intended for use in the manufacture of a drug product, including those that may not appear in such drug product. FDA May 2023 guidance also said high-risk drug components are components that, through historical experience, have been found to be at a higher risk of EG-DEG contamination compared to other drug components. So a high-risk component that is intended as an excipient or other component of a drug product is a drug as defined by Section 201G of the FD&C Act. So excipients are also drug components. They also need to be in compliance with the CGMP requirements. So here is the 21 CFR 211.84 CGMP identity requirements. According to this CGMP requirements, each lot representative samples of each shipment of each lot shall be collected and tested. And also, at least one test shall be conducted to verify the identity of each component. So according to the CGMP identity requirements, so if the EG-DEG testing is in the ID section, so this test needs to be tested for each lot. To summarize the FDA May 2023 guidance, the similarities of cases are that DEG and EG contaminated components entered the pharmaceutical raw material supply chain. However, drug manufacturers did not perform full identity testing and solely relied on the certificate of analysis provided by the supplier. Additionally, the CLC was not from the original manufacturer, nor did it disclose the origin of the excipient. So supply chain is important. According to the regulatory requirements, manufacturers of finished drug products must also comply with the CGMP regulations codified in 21 CFR parts 210 and 211. Testing bulk or repackage the high-risk components of DEG and EG content is consistent with the CGMP requirement and the section 501A2B of the FD&C Act. A drug, including a drug component with a name recognized in the USPNF, must comply with compendial identity standards or be deemed adulterated, misbranded, or both. So you can see the keywords for the guidance are the identity and the supply chain. Unfortunately, some companies still do not follow the FDA guidance and do not follow the CGMP requirements. FDA issued more warning letters this year. On the right side, you can see an example warning letter. For this form, they failed to test the identity and the conformity with all appropriate written specifications for purity, strength, and quality. For glycerin, the company failed to adequately test incoming components at high risk of diethylene glycol or ethylene glycol contamination for identity before using them to manufacture your drug products. I hope this workshop will make more companies be aware of these CGMP requirements and also the FDA guidance. So next, let's talk about USP's perspective and activities. This slide summarizes USP's efforts on DEG and EG testing for high-risk excipients. So back to 2007, USP received the first FDA letter regarding regarding the inclusion of EG-DEG test in the ID section of glycerin monograph. In 2009, USP received another letter regarding propylene glycol and the 4 sorbitol solutions. So between 2007 to 2010, USP has collaborated with stakeholders, including the FDA, to include the EG-DEG ID test in these 6 monographs. And from 2011 to 2013, USP also collaborated with stakeholders to include the EG-DEG ID test in hydrogenated starch hydrolysis. All these 7 monographs have been included in the FDA May 2023 guidance. And from 2013 to 2014, USP developed a new general chapter 469 to analyze EG-DEG and TEG in ethoxylated substances. Due to the most recent incidents, USP published a DEG-EG toolkit to help stakeholders to address this important issue. This toolkit includes relevant monographs, chapters, and other resources. And it is a free resource for all stakeholders who are interested in this testing. And currently, USP is collaborating with the FDA lab and also stakeholders to develop appropriate EG-DEG ID tests for different packs. So as you can see, USP has been working with FDA and the stakeholders to address this important public health issue for decades. So USP has already included the EG-DEG test in the ID section in these 7 high-risk excipient monographs already. So the major purpose for the update is to address the economically motivated adulteration. On the other hand, USP also developed this general chapter 469. There are 17 excipients listed in this chapter, and this gas chromatography FID method is to analyze the EG-DEG and the TEG process impurities for these excipients, because they are all manufactured using ethylene oxide as a starting material. I want to let you know that this chapter 469 is a standalone analytical testing procedure-based chapter. It does not contain acceptance criteria. Recently, USP also proposed a new general chapter 470 to propose several EG-DEG test methods for polyethylene glycol only. That chapter is also a standalone analytical testing procedure-based chapter. I will talk about it later. So what are USP's current challenges and activities? As I mentioned, USP has already implemented the EG-DEG test in the ID section for those 7 high-risk excipient monographs already. Currently, we are focusing on the polyethylene glycol monograph update. Because on February 10, 2023, USP received another letter from the US FDA that requests to include the EG-DEG test in the identity section of excipient monographs, including polyethylene glycol and the polyethylene glycol 40 castor oil. So the current polyethylene glycol NF monograph has no identification test. In the impurity section, there are 2 EG-DEG test methods for molecular weight below or equal to 1,000. One is a packed GC column method for molecular weight below 450, and another method is a UV method for molecular weight from 450 to 1,000. And I also want to mention that the NF polyethylene glycol monograph covers molecular weight from 200 to 8,000 USP also has another separate USP polyethylene glycol 3350 monograph because PEG 3350 is a dual active. USP also plans to develop a separate new monograph for polyethylene glycol 20,000. So there are two major challenges. One is methodologies. For molecular weight below 1,000, the packed GC column method are difficult to purchase now. And for the UV test method, it has a tedious procedure, less accuracy, lack of capable analysts to perform the test. And for molecular weight above 1,000, there is currently no EG-DEG test method in the monograph. Therefore, we are collaborating with the stakeholders to develop a new capillary GC method for molecular weight below or equal to 1,000, and also to develop a new gel permeation chromatography method for molecular weight above 1,000. Another challenge is acceptance criteria. The current PEG monograph has not more than 0.25% of combined EG and DEG. However, in FDA 2023 guidance, it has not more than 0.10% of EG-DEG for EG and DEG respectively. Based on the information and feedback from stakeholders, they have concerns about the liquid low molecular weight PEGs to meet the acceptance criteria for not more than 0.10% of DEG. So the reason is polyethylene glycol is manufactured by polymerization. So if the manufacturing process is not complete, more unreacted EG could be detected. Therefore, as the PEG molecular weight increases, the amount of EG-DEG decreases. USP engaged the stakeholders to provide batch data of EG-DEG levels in different PEGs, especially for liquid and semi-solid PEGs. USP published a general announcement along with a short survey last year, and 127 stakeholders browsed the survey. Based on the 64 responses completed for crucial questions, very useful information and feedback were received. The EG-DEG batch data provided by stakeholders for different PEGs helped USP setting appropriate specification limits for EG and DEG. In addition, stakeholders also provided several in-house EG-DEG test methods. So they help USP providing available EG-DEG test methods to evaluate the suitability for their intended use. So that is a great collaboration between stakeholders and the USP, and USP appreciates the support of stakeholders. Based on the information received, the USP expert committee proposed to use a stepwise approach. In step one, revise the polyethylene glycol monograph to include the existing 469 EG-DEG test in the ID section via an accelerated revision process. In step two, USP proposed a new general chapter to propose available EG-DEG test methods. So the polyethylene glycol monograph revision proposal was published in PF 50 issue 3 from May 1st this year. The 3-month public comment period ended on July 31st. Due to the comments received, USP plans to have a delayed implementation for this proposal. Please stay tuned for the upcoming general announcement. I will not talk about the details. You can find the information on this slide or log into a Pharmacopoeia forum and to check the actual proposal. And in addition, a new general chapter 470, determination of ethylene glycol, diethylene glycol, and triethylene glycol in polyethylene glycol has been proposed in PF 50 issue 5 from September 1st. So it is currently in the 3-month public comment period. So please evaluate the method and provide your feedback. 4 new testing procedures have been proposed in addition to the existing 469 GCFID method. This chapter is also a standalone analytical testing procedure-based chapter without acceptance criteria. Stakeholders can evaluate the equivalency and the interchangeability of these methods and use them as alternative methods as necessary. So next, I want to talk about Pharmacopoeia discussion group PDG harmonization on these related high-risk excipient monographs. So PDG was established in 1989 with the member of European Pharmacopoeia, Japanese Pharmacopoeia, and US Pharmacopoeia. India Pharmacopoeia joined the PDG last year, and WHO has been an observer since 2001. The monograph, the glycerin, propylene glycol, polyethylene glycol monographs are under the PDG work plan for harmonization. So for USP, as I mentioned previously, we have put the EG-DEG test in the identification section for molecular weight below or equal to 1,000. For molecular weight above 1,000, we have a gel permeation chromatography method in the impurity section because solid packs have less risk of adulteration. And European Pharmacopoeia has EG-DEG test method in the impurity section. They also have different specification limits for EG and DEG for the propylene glycol and the polyethylene glycol currently. And the Japanese Pharmacopoeia also has the EG-DEG test in the impurity section. They have the same acceptance criteria for EG and DEG in all 3 monographs. They are the same as the USP currently official monographs. And the India Pharmacopoeia has the EG-DEG test method in the impurity section for glycerin and the propylene glycol. Currently, they don't have any EG-DEG test to control the EG-DEG level in polyethylene glycol. But PDG is working on the harmonization of these 3 monographs, and hopefully we can also harmonize the EG-DEG test. Even if different pharmacopoeia place the test method in different sections, and the testing procedure and the acceptance criteria, hopefully we can harmonize those. So for the next steps, currently our expert committee is addressing the comments received on the polyethylene glycol PF50 issue 3 proposal, and we will publish a general announcement with the delayed implementation of this proposal. Please keep an eye on the USP website on this for this upcoming general announcement. And regarding the new journal chapter 470, please evaluate the methods and provide your comments. And USP is also collaborating with the FDA laboratory on the evaluation of available EG-DEG test methods for polyethylene glycols. PDEG is working on the harmonization of glycerin propylene glycol and polyethylene glycol monographs. USP is the coordinating pharmacopoeia for glycerin and the polyethylene glycol monograph harmonization, and the EP is the coordinating pharmacopoeia for the propylene glycol monograph harmonization. So you can download the EG-DEG toolkit through the link on this slide, and also the excipients listed in the current EG-DEG toolkit is shown here. I want to let you know the propylene glycol, the EG-DEG toolkit will add more information. For example, propylene glycol, polyethylene glycol, 40 castor oil monographs will, and several other excipient monographs will be added to the toolkit soon. And for the related reference standards, USP provides diethylene glycol, ethylene glycol, triethylene glycol reference standards, and also the internal reference standards for the GC test, and also the reference standards for these high risk excipients. We have hyperlink on the slide, so you can click those the links to the USP store. And in summary, collaborations. I repeated the important things 3 times. Regulators to enforce the regulations, pharmacopoeias to ensure up-to-date standards are available, and the industry to comply with the CGMP requirements by implementing EG-DEG related compendium quality standards. At last, I want to mention that USP is currently recruiting expert volunteers for the next cycle. You're welcome to scan the QR code to find more information on the USP website, and also apply for the volunteer role to utilize your expertise for public health standard setting. Yeah, these are the, if you have questions, please send email to nfmonographs@usp.org. I also included my colleague Ila's email address on this slide. So please contact her if you have questions. Now, I will be happy. I'm very happy to answer your questions next. Thank you.
Thank you, Dr. Jenny Liu, for the insightful presentation. Okay, we will move into Q&A session for 15 minutes. Ladies and gentlemen, you can raise hand through the Zoom meeting, or you can type in on the chat box of slido.com. Yeah, please. If you have a question, can raise hand in the Zoom. Okay, Mr. Dr. Janilu, maybe we read question on the chat box or slido.com.
Okay, so I want to so to answer the question, the first one, if we have 10 containers with the same batch lot coming at the same time to our warehouse, is it okay to only test one container for EG-DEG contamination? Unfortunately, I think according to the FDA guidance, and each lot and each container should be tested, and especially one FDA director who answered this question specifically, and he has data showing that for each container, you must test each container, even the composite samples is not allowed. And the second question, so do I answer your question? So the second question is, can we use risk assessment to prioritize, determine the importance of
Yang
EG-DEG
pertamanya
testing
juga.
based on our analysis trend of our excipients? So regarding the high-risk excipients in the FDA guidance, I think the EG-DEG test needs to be tested to be in compliance with the CGMP requirements and FDA guidance. Regarding other excipients, if you have the risk assessment to demonstrate there is no chance of EG-DEG existing in your excipients, and you have a record showing that, and you can contact FDA to get their feedback and to make sure it is fine. That's a suggestion. What do you think about EG-DEG TEG test method for tag about 1,000 in Chinese pharmacopoeia? Is it reliable to use? Unfortunately, I cannot comment on that, but I mentioned that in the tag monograph revision proposal in PF 50 issue 3, we have a gel permeation chromatography method. You can evaluate that. And also in the new journal chapter 470, we propose several EG-DEG test methods. You can also evaluate those methods for the suitability for your intended use. And if USP intended to introduce a general chapter for ethylene glycol and diethylene glycol determination or solution drug products. You have very good question. Actually, it is very challenging to develop a universal test method for oral drug, oral solution drug products, because different drug product has different matrix. The method is really difficult, challenging to demonstrate the specificity of the method. And also, but currently, I think some stakeholders contact us, and we are considering to develop some method. And also, if you have any method for the EG-DEG testing in drug products, please submit it to USPNS. Our EC will discuss those. Yeah, let me know if my time is up. And what's the next question? What is the regulation in US regarding EG-DEG testing in finished product? If we already tested in all material, is it still required in finished product? Yeah, this is a good question to talk with the FDA. I think finished drug product needs to be tested. However, if you can demonstrate all your raw materials are free of EG-DEG, and you also have the risk assessment, and you provide all those information to FDA, FDA may consider it. But I recommend you contacting FDA directly. And I think I answered that question already.
Doctor,
And another, yeah.
We will read question on the chat box. Okay. From EG-DEG from Conimax. In the future, is USP intend to produce a general chapter for ethylene glycol and ethylene glycol determination in all solution drug product?
So
maybe you can show in box chat.
So which question are you talking about? Because there are several questions. I'm sorry, I did not follow you. So should I continue to answer the questions, or Ella will help summarize all these questions and pass it to me? We can answer it offline. So what's your
Can you open the chat box? There's 2 questions that we received on the chat box.
Chat box. Okay. In the future, if USP intended to introduce a general chapter, I think I answered that one.
Yeah, I think you need to answer the second one.
Yeah. May I ask about EG-DEG in the excipient, for example, in upper dry that contains tag 3350 upper dry coating in the tablet is upper dry must be tested. I think for the raw material release testing of tag 3350, EG-DEG needs to be tested. In the current tag 3350 monograph, there is a EG-DEG test method there, which is a gel permeation chromatography method. If EG-DEG is an ID test, it means we should test the ID for each container. Example, 1 lot consists of 3 containers. I think I answered that question also, because it should be tested for each container. Yeah. So how about time, Ella? So should I? So the next speaker to.
Okay. Thank you, Dr. Jenny Liu, for the wonderful session. Our 4th question cannot answer. My team will collect and will send to Dr. Jenny to answer. Thank you very much, Dr. Jenny Liu, for the wonderful session.
My pleasure. Yeah.
Okay, ladies and gentlemen, now let's move to our second speaker, Dr. Karen Love from Australia TGA. Dr. Karen, okay, yes.
Hello, good morning, everybody. Let me just bring up my slides.
Introduce my let me introduce yourself, Dr. Karen. Dr. Karen Love is a chemist by training, obtaining a PhD in synthetic organic chemistry at the University of Melbourne. Her research involved preparing sulfate carbohydrate substrates to investigate carbohydrate sulfates, a group of enzymes responsible for a series of metabolic diseases. After obtaining her PhD, she spent several years as a postdoctoral researcher at the Walter and Eliza Hall Institute, where he investigating novel anti-cancer treatment targeting BCL2, a regular protein that blocks cell apoptosis, leading to some cancer. Karen then moved to the TGA and has over 10 years experience in pharmaceutical chemistry evaluation for prescription medicines. The team responsible for chemistry and quality control assessment for application for new medicine, new chemical and generics and variations. Karen has spent several years as a key decision maker for new and various medicine and has been at the forefront of several of TGA's response and participation in different international working groups. Karen now was in TGA's regulatory strengthening program, making use of her extensive evaluation and broad regulatory knowledge to provide technical and regulatory strengthening assistance for countries across Southeast Asia and the Pacific. Dr. Karen Love will be presenting about regulatory perspective on quality assessment of drug product with respect to the EG-DEG as well as elemental impurity. Please, Dr. Karen Love, and you have 30 minutes to present the presentation. Please.
Okay. Thank you, everybody, and thank you very much to the organisers for inviting me to present today. So I hope that you can all see my slides there. So today I'm going to be providing a bit of a regulatory or regulator's perspective of the impurity control. So next. Oops, apologies. Okay, so I thought I'd just briefly start off with a bit of a regulatory overview on general impurities in medicines and ingredients, and why, as a regulator, we need to be worried about them, the risk and the safety that comes from those particular products, and ways that control strategies can be applied and put in place to minimise the risk and potential safety challenges with these types of impurities. And then finally, I'll sort of close off with a bit of a discussion around regulatory approaches and challenges in changing requirements to quality controls and impurity discoveries. So first, as we all know, medicine ingredients can affect the drug product performance, quality, and safety. Of course, we have various types of ingredients in medicines, which Dr. Jenny nicely summarized for me in the earlier slides. And excipients, or non-active components of medicines, can often make up a very large portion of the medicine that's being administered to the patient. So they're really important factors to consider when a product is being developed and when it's proposed to be approved for supply and it's really important to consider what impurities can come in from these components. So the quality control measures for each excipient need to be considered and based on the risks with the specific product. So we're talking about risks that not just might cause changes to product performance, but of course risks that come about from impurities and contaminants within individual components within a medicine. So different in excipients and ingredients will have different risks associated with them. Ingredients and excipients can come from very different and wide-ranging sources. And in Australia, we expect that the market authorisation holders know what risks their medicine, which includes the excipients, all ingredients, what risks may be present in the product to the quality, safety, and function of each of those ingredients that are used in the medicine. So of course, there are different risks associated with different types of ingredient sourcing, and I just have a few slides here to very briefly go over those. So ingredients can come from vegetable sources. They can be derived and isolated from natural vegetable type products. There can be a risk of all sorts of impurities that come about from these particular ingredients. You can have toxins, microbial content, heavy metals, pesticides, depending on how products are grown. And these factors are important to consider in the risk posed to a product or a patient when an excipient from one of these sources is used. There are mineral-based ingredients and excipients, so where they're derived from mining, for example. Impurities within these types of ingredients can be those directly co-located in the mining location where the ingredient is being sourced or its purification processes. Ingredients can of course be synthetically made. These risks can come from contamination of the manufacturing processes. Maybe there are inadequate in-process controls, insufficient controls of starting materials that are being used to manufacture ingredients or components of a medicine. There can be sometimes, in the case of elemental impurities, the deliberate addition of catalysts, which are important for the production of these particular excipients. There can be residual solvents and other types of impurities present in products that are made via a synthetic route, and whatever those impurities are will depend on the synthetic route and the controls applied to a manufacturing process by a manufacturer. And this is, of course, also goes for excipients, not just drug substances, which is what we commonly think about. And of course, there are other challenges or potential risks associated with certain ingredient types. Certain microbial risks might be higher depending upon the source. High-risk excipients, such as those which are going to be used in sterile products, need to have additional considerations around sterility aspects and bacterial toxins, for example. So these factors are known to come about from the source that the material or the process that the materials are being isolated or sourced from. There are also risks associated with manufacturing and storage challenges. So things that can come about through cross-contamination during manufacturing processes. Maybe there's poor cleaning practices, for example. The complexity of the supply and the distribution chain, and our previous speaker mentioned some of those challenges and having a clear understanding of what the supply chains are like. Packaging integrity and tamper-proof evidence of packaging, of course, may be factors to consider in potential other risks associated with product and ingredient quality. Product stability is another factor, and appropriate storage conditions and containers. So there's the chance that if inappropriate conditions or storage conditions or containers are being used, then that can create or lead to the formation of more impurities. And of course, there's the potential for adulteration and contamination. Now we know there have been lots of unfortunate cases recently and throughout history around DEG-EG, and we are aware of some products that do have a known history, other products that have a known history of potential contamination issues or purification challenges. However, they've come about. And the results of those have quite high safety risks associated with them. So for example, of course, there's the DEG-EG in various excipients. There's potential viral contamination from some animal sourced materials. We know that there are some high risk solvents that can be present in other solvents, such as benzene in ethanol and methanol in ethanol, for example. So there's lots of factors to consider when, from a product development perspective, a product safety and maintenance perspective, and a regulator's perspective in thinking about how we manage these risks when we're thinking about the appropriateness of excipients for supply and for use in medicines. So we at the TGA apply lots of ICH guidance documents in the way that we approach our regulatory perspective. And the ICH uses a risk-based approach to focusing on impurities in this particular case. Impurities based on both their toxicological and safety profiles, but also the route that the products are intended to be used. So in the case of elemental impurities, the ICH has come up with a list of different classes of elemental impurities which are not to or should be reduced or restricted for inclusion in medical products. And these classes are based on risk, and this risk also considers the route of administration. So a lot of what I'm going to focus on now is on elemental impurity aspects, but of course there will be some crossover with the requirements that we have in place around diethylene glycol, ethylene glycol, and the considerations are also relevant. So just bear with me, I will be talking about elemental impurities, but of course, like I said, a lot of these factors and considerations are relevant as well to ethylene glycol and diethylene glycol. So elemental impurities can arise from numerous different sources. They can come about from intentional addition. Like I said earlier, there are ingredients which are commonly synthetically manufactured, and in order to do that, these processes require the addition of elemental impurity catalysts, for example, to drive that reaction. Sometimes they're used in reagents or processing aids in order to effectively produce a particular ingredient, be it a drug substance or an excipient. They can come about through contaminants in the raw materials that are used to produce those particular ingredients in the medicine. And they can also come from extraction or leaching of the manufacturing equipment or container closure systems that the products are stored in. And the reason why ICH and the TGA have adopted ICH recommendations around the risk-based approach to the control of elemental impurities is that there are some elements which have no therapeutic effect at all and are known human toxicants. So elements such as arsenic, cadmium, mercury, and lead shouldn't be included in products at all. Whereas there are some others that, although not having a therapeutic effect, are sometimes required or present naturally, and that there can be small amounts that can be tolerated in products intended for human use. So the ICH has outlined a risk-based approach and a risk assessment, sorry. The ICH outlines a risk assessment that should be used in order to consider whether elemental impurities are present in a product. We expect that this information is communicated to us as a regulator from our market authorization holder, from the manufacturers in turn, to provide us with information around levels of potential excipients or elemental impurities in products. The risk assessments describe the way or suggest a way that the assessment should be performed, things or aspects to consider around the manufacture, formulation, storage of a particular medicinal product, and also provides some permitted daily exposure limits, so limits that we can use to set appropriate limits for elemental impurities. So things like considering the ingredients that are being used, how much of a particular ingredient is used in a particular formulation, what equipment are being used, what's the manufacturing process, and how is the product stored before it's administered to a patient. And so again, coming back to these elemental impurities are exactly that. They're impurities. They provide no therapeutic benefit, and just the same as every other impurity, they should be controlled within acceptable limits and the appropriate investigations conducted in order to make sure that the control strategies in place are appropriate. Things to consider in the risk assessment are outlined on this screen here, and this is taken straight from the ICH. There are multiple different information sources that can be used. Apologies, my light's just gone off. There are multiple different ways in which this risk assessment can consider information and obtain information to inform a risk assessment and a risk control strategy. So there can be prior knowledge around the natural abundance of particular excipients. What concentration can they be typically found in? What literature are available? Of course, a supplier of an excipient or a raw material should be aware of the quality of the product that they're supplying. There should have some information around the efficiency or the potential for there to be elemental impurities in a particular component or raw material or excipient, and the ability of their particular manufacturing process or purification processes to remove those impurities. Of course, testing is an important factor to consider where there is a risk, and again, the overall composition of the drug product is an important factor to consider as to whether then the level of control required for the drug product. So the elemental impurities in the drug products can come from multiple different sources. It can come from every raw material or any raw material in the manufacturing process to a component of a medicine, be that a film coating material, the ingredients in a tablet, or water used for an injectable product. Every component of that particular product has the potential to introduce risk of unwanted contamination or impurities present. And each of these components need to be considered in addition to container closure systems, manufacturing systems, and materials that are in place, as well as components that are used to assist in the manufacturing process. So risks can be reduced in making sure that there's really good knowledge and understanding of the processes, the controls, and the ingredients and the quality of those ingredients that are being used in a medicine. Having good understanding of processes, what equipment are being used, how those equipment are qualified, cleaned, for example, as well as, of course, good manufacturing practice processes are really integral to ensuring that ultimately the product quality is appropriate and well controlled. It's also making sure that the excipients are monitored, or processing aids are monitored, and making sure that they're applying with the appropriate controls, as well as, of course, the GMP requirements of having good understanding of all those excipients that are being used and ensuring that GMP practices are maintained and consistent throughout the process. The ICH guideline talks through different ways that control limits can be designed or proposed, and the TGA will accept both of those, where a maximum permitted daily exposure can be determined based on the daily dose of a component. So it can be considered, but should be considered as a whole in terms of the drug product. So it's important that the complete and full formulation and composition of the product is known, as well as there's a slightly more conservative approach, which the ICH itself has done some calculations on. And either of those approaches are fine, depending on whether the appropriate control strategies are in place to justify a particular limit. Now, contaminants are specifically excluded from the ICH guideline, and that's because they're contaminants. They shouldn't be in a product at all. So there's no real regulatory response or need for them. Well, there's no need for them to be in the product, and in fact, they shouldn't be there at all. So they're more appropriately addressed through GMP practices. But there are some practices or requirements that, as a pre-market, from a pre-market perspective, that can be included and considered as part of setting up an appropriate market authorization package or market authorization approval. So there's ways that both GMP and pre-market or market authorization function together to ensure appropriate control and appropriate quality of products supplied. And the ways that these potential contamination products should be monitored and risk assessed are very similar conceptually to what we've just went through for elemental impurities. There should be good process understanding. What equipment are being used? How well have they been cleaned? What quality requirements are the excipients claiming compliance to? Are they claiming compliance to a compendial excipient requirement? Have they been demonstrated? Can they demonstrate that they do meet that? Does the manufacturer have good understanding of the processes used by the suppliers to manufacture each individual component of a product? Do they have an appropriate selection process in choosing those suppliers? Do they routinely qualify and re-qualify their suppliers? What are the relationships like? Is there adequate information transfer from a supplier site to a product manufacturing site? And all of these things are considered and required under good manufacturing practice processes. In thinking about particular excipients that have known risks of contamination, and especially those with contamination with products that are of a safety risk, then we should be applying even closer scrutiny to these particular excipients, both in the GMP practices, that supplier qualification, testing, and relationships, as well as the market authorization practices. And that's, I guess, brings me on to the approaches applied by the TGA and our regulatory expectations around good control of ingredients and impurities in those ingredients. So as I sort of mentioned before, a lot of our approach comes down to a joint approach between our GMP teams and our market authorization teams. Obviously, our GMP teams will do their inspections based on current good manufacturing practices and will consider any market authorization approvals that they might have in place at that particular manufacturing site, as well as the controls applied to the manufacturers and the manufacturing supply chains and controls applied to suppliers. And from a pre-market perspective, we can look at this information in terms of the controls applied to each of the components that are used in a medicine, including packaging and other raw materials. So from a pre-market perspective, we expect that an application will state the function of each excipient. It'll state the quality standard of each excipient that's going to be used before it can be used in manufacturing, and the pharmacopoeial standard that applies. We have a default requirement that all ingredients will comply with a pharmacopoeial monograph where there is one. And we expect that there's a risk assessment done of those particular excipients. Are any of these excipients high risk? Do they have a particular critical function on drug product performance? And do they have a particular critical function or potential critical function or potential critical quality attributes that might affect product safety? And that's part of the regulatory considerations as well. The market authorization holder needs to be aware of all of this information and provide it to us. And the manufacturers need to be able to provide us with the same evidence that their ingredients that they're using comply with the required specifications and meet appropriate quality controls. How they go about providing us with that information will differ depending on the nature of each excipient. But ultimately, what we're looking for is to make sure that a manufacturer has a good knowledge of their manufacturing processes, their excipients, the manufacturing lines, their suppliers, everything that I've discussed previously with the risk assessments, and how all of this information forms part of an appropriate control strategy. What controls does the manufacturing site have in place around all of these parameters? How are they going to make sure that they're effectively manufacturing, controlling, testing as necessary, the quality of the product that they're manufacturing? We have expectations, of course, that the products that they're supplying are high quality and safe to our populations. So we need to make sure that that is demonstrated to us, and we need to be confident that that information will be able to be maintained throughout the product life cycle. It's not just an initial product registration or approval requirement, but it's an ongoing requirement to make sure that the products remain safe and the ingredients that they're using also are safe. So there's lots of different ways, like I said, that they can provide us with that information. It'll be part of the entire dossier that they provide to us, and it'll be a discussion on the control strategy of impurities in each component of a product. So we've discussed before around the risk assessment considerations for elemental impurities and also ethylene glycol, diethylene glycol potential contamination and the ways that those risks can be reduced. And I've just reproduced that information here on the slide. But we also then need to think about other ways or other potential sources of contamination. Specifically focusing on elemental impurities, those considerations are around, well, what's the route of administration? What's the risk to the patient? Is anything potentially added or likely to be present based on a manufacturing route of a raw material? Is there anything intentionally added in terms of the manufacturing process of a drug substance, for example? What's the likelihood of leaching and a potential interaction? And if there is a potential for an elemental impurity or DEG, EG, to be present in a raw material, an excipient or the drug substance, if there is that potential that exists that it could be there in the drug product, then what's the control strategy that the manufacturer will apply to the product? They need to describe that to us. They may need to perform some testing if there is an identified risk of elemental impurities, for example, being in the drug product. They may need to set up control strategies and justification approaches, and they may need to include limits in a drug product. So it all forms a larger part of a broader story that each component used in a drug product needs to be considered individually and then again as a whole for the drug product. And this is the same approach, like I said, for any other ingredient used or any other impurity that may be potentially present in a drug product. So finally, just to close off, I thought I'd just briefly talk through some of the approaches that the TGA has gone through in adopting some of these requirements, specifically around products that may already be on the market when we're introducing these sorts of requirements. So thinking about elemental impurities, there was a fairly long transition period included in the ICH guidance document that allowed manufacturers to do the appropriate investigations and identification and testing as needed of particular products. And that was definitely implemented and allowed here at the TGA. And in fact, it was several years later that we formally adopted the ICH requirements, although we had been informally requiring those investigations to be provided well before that. And during this implementation process for new products, we also allowed pre-supplied or pre-approved products to also go through this gradual update and provision of information to the TGA through our normal variations processes. And that was this time frame and transition period was considered and allowed based on a risk-based approach. We had already been assessing drug substance and drug product controls of products based on scientific information provided to us in the dossier. For example, where we knew that catalysts were intentionally being added during manufacture, we were already requiring that these particular elemental impurities were controlled appropriately at some point in the manufacturing process. There had been instances in the past where certain active ingredients like bisphosphonates or zoledronic acid, for example, were known to extract elemental impurities from glass. And so we already had some controls in place and required by the TGA before these particular elemental impurity requirements for the drug product were introduced. A really important thing to consider is how ongoing product supply will be maintained in these finding potential products that may have, for example, these impurities in them. There's always it's a risk-based approach. We have had to manage potential shortages due to important medicines with small deviations to elemental impurity contents, for example. We've had to allow and manage to maintain the supply of these important medicines, even though there have been some challenges in reducing elemental impurities. So it's an ongoing, and I think it was very nicely summed up by in the previous presentation, that it's all about collaboration. Manufacturers and market authorization holders and regulatory agencies need to work together to manage the supply of critical medicinal products, but yet balancing, of course, the safety risks associated with that. So it's a very much case-by-case basis, but it's important to consider how or when or if these factors may be able to be how you will adapt to these particular challenges if cases are observed post implementation. So really important things to bear in mind about in maintaining appropriate medicine supply where the risk can be mitigated. And of course, we've had some other recent examples of these very similar types of approaches of having to manage product supply and risk. So for example, the nitrosamines, an issue was identified, a contaminant was identified. There were some critical safety issues. Some products were required to be recalled due to safety challenges, whereas others perhaps required a slightly different approach. So there's a need for some regulatory flexibility in being able to manage medicine supply and patient safety. And it's important that all of us, everyone is working together to make sure that patients are ultimately being provided with products that are of highest quality whilst maintaining appropriate product supply. So just some final food for thought or things to think about in implementing requirements and introducing new requirements to products already on the market. So that brings me to the end of the presentation. Thank you very much. Any questions?
Yeah, thank you for Dr. Karen Hoff for your insightful presentation. Now we move to Q&A session. Please, ladies and gentlemen, if there are questions, you can raise hand in Zoom or type on the box chat or slide.com. Yeah. Okay, there is question from Wildan Alfian Nur. Is there any EG-DEG limit which required by TGA currently?
Good question. We haven't published separate information or guidance in relation to DEG, EG. So we're currently using the requirements that are applied by other regulatory authorities, as well as those included in specific monographs.
Okay. Ibu Ibu Nina, you raise hand. Please you say your question to Dr. Karen. Oh, okay. I will chat. Oh yeah, please, Ibu Nina. Yang mana? Yes. Ibu Nina.
I see there's the question in the chat box. Shall I just
Okay.
answer that one?
Yes.
It's about residual solvents
Yeah.
in the manufacturing process of an active pharmaceutical ingredient. So is the residual solvent required to be tested in the finished product, is the question?
Yes.
Yep. So we take a risk-based approach to that. So it will depend on the risk assessment that's been performed or the control strategy that's applied to the product. I think the control strategy is a nicer way to think about it. So it will depend on if the active pharmaceutical ingredient is being controlled itself for residual solvents. We would expect that if an API is manufactured with residual solvents, then it will be controlled for residual solvents or demonstrated what those residual solvent levels are. Now, if we think that the API is well controlled and that the finished product manufacturing process does not introduce any more risk of adding residual solvents, then it may not be required to be included in the finished product testing or the finished product specification. So it will depend on the understanding of the manufacturing process that the manufacturer can provide to us, as well as subsequent steps that are being used to produce that finished product.
Okay, thank you, Dr. Karen. So we next to question on slido.com. Please, my team share the question. Okay, this question in slide.com. For drug manufacture, we have a limited access and detail to actual production on the drugs. So what kind of approach to gather information for this assessment?
So that's a good question. So part of the requirements that we have at the TGA is that the manufacturer is aware of all of this information and that they can provide us with that information if we ask for it. Sometimes it's required to be provided to us as part of a registration application or an application for approval. So all of this information should be able to be provided to you by a manufacturer that has the good relationship and knowledge around the excipients and the raw materials that are being used, in particular the drug substance that's being used as well. Any component that's being included in that drug product should be part of the finished product manufacturer's knowledge. So if it will depend on the particular scenario or the confidence that you have in the information that's being provided to you. If you don't have enough information and access to the details around the production of a drug substance or the quality of an excipient, then what sort of information does the manufacturer have and how confident are you that they have access to that information as well? So it's a decision, I suppose, to think about how well do you feel that that information is known. So like I said, we expect that our manufacturers have access to all of this information and are aware of the quality controls and synthetic routes used, particularly for drug substances. The synthetic routes are used to manufacture components of the product. So that might be something to consider. Perhaps if you're not getting that information, then in order to make a decision, perhaps that information some information is required around those particular routes.
Okay, thank you, Dr. Karen. Next question in Slido. Yes, to perform risk assessment on elemental impurities of finished product, are the testing result of finished product mandatory as one of supporting data?
Also, good question. It forms part of the overall information package that you have. What's the control strategy that's being applied to a particular component? So the ICH guideline does run through a few ways to consider how best to perform a risk assessment and how best to decide when testing should be performed. And it sort of comes down to if there's a chance that elemental impurities or likely or may be present in the product, and there's no appropriate control strategies perhaps further back in the process, then perhaps testing will be required, but it will depend on, for us at the TGA, it will depend on that larger package of information that we've been provided with around the routes of manufacture and potential controls that are applied to each of the raw materials and intermediates in that manufacturing process. But if we didn't have access to any of that information, then perhaps it might be more relevant for us to think about requiring finished product testing. But a risk with that is that you don't have the opportunity to look at the larger package of information. How confident are you that the manufacturer has good controls in place, good measures in place, good GMP in place to make sure that what you're actually seeing in those testing results is representative of the product that's being supplied to the public?
Okay, thank you, Dr. Karen. The next question we'll get from chats from Wildan Alfianur. It means that like example that the API manufacturer already established better control for the residual solvent and controlled via API test before use for production. Then we need also consider any residual solvent from other excipient. Then how if some excipient do not have monograph reference, for example, flavor or colorant? How we assess the residual solvent from this RMS, or we can bring the residual assessment directly to the finished good.
Yeah, it's a good scenario. As you've said, if the API is already controlled for residual solvents and controlled in that API, then yes, you will then you'd need to consider whether any residual solvents come from other excipients as to whether another control is required in the finished product, because you're adding more risk and more chance that residual solvent may be present in the product, again from an additional step that's being used. If there are excipients that don't have monograph references, like flavorings and colorants, for example, how do you know how to assess residual solvents from these particular materials? Well, that comes down to, I guess, having that information from the supplier. The supplier should know what the quality controls are required for that particular raw material, excipient, colouring agent, whatever it might be. Your supplier will, like I said, should know the manufacturing process and the way that it's been produced, and so should have a good knowledge of the quality controls. And so if you should be able to ask for that information from the raw material supplier. Do you use any residual solvents, for example? Do you add any elements? What impurities are coming from these particular raw materials that you as a manufacturer, that the regulator in turn would like to know about? And you need to, you should be able to get that information and include that in your risk assessment around whether a control is required, testing might be required.
Thank you, Dr. Karen. Dr. Karen, this is the last question of your Slido. Is if the analysis result is the finished product below PDA but exceed the threshold 60% PDA, is it necessary to carry out routine control per batch?
I think that's what the guidance document says, and it may be that if these are the results that you're observing, it may be necessary to carry out routine control per batch, but it might also be acceptable from a regulatory perspective that you introduce a control at an earlier stage during the manufacturing process, for example. For example, if it's an API, an active pharmaceutical ingredient that uses an elemental catalyst, then perhaps it may be appropriate to introduce a test immediately after that particular synthetic transformation to control the elemental impurity content rather than in the final API. So where that control occurs will be up to a particular manufacturer to decide, but it should be somewhere if it is consistently above the threshold in the final API.
Thank you, Dr. Karen, for this session. For question have not been answered by speaker, we will collect and send to speaker. After get answered, we will send all to you. Thank you very much, Dr. Karen.
Thank you. My pleasure.
Okay, ladies and gentlemen, now we move to last session. We will have our speaker for today, Dr. Christian Zen from USP Europe. Okay. Good afternoon, Dr. Christian. Allow me to introduce yourself. Dr. Christian Zen is Senior Manager in the Scientific Affairs Group for the EMEA region, with a focus on small molecules, USPS General Chapter, and biology. Dr. Zen collaborates with scientific experts and stakeholders and is responsible for protecting and growing USP scientific reputation in the region and globally. Before joining USP, Dr. Zen worked for 17 years in the field of pharmaceutical reference standard with a focus on impurities. And before that, in the IVD, in vitro diagnostic industry, his scientific expertise includes impurity testing, reference standard characterization, and adjacent fields. Dr. Zen has published several articles and white papers on topics such as impurities, overview of certified reference material, and the use of reference standard in method development and validation. Dr. Christian Zen will be presenting about elemental impurities test. Please welcome Dr. Christian Zen. You have 30 minutes to present your presentation, please.
Okay, thank you, dear Elsa, for the nice introduction. I will talk a little bit low. I hope you can all hear me. And I also apologize for the light that you see here and the somewhat dark side here, because it's still dark here in Austria, where I'm presenting today from. It's just 6:20, and we are coming to autumn and to winter, so it's still dark. And I'm also sitting in a hotel room and don't want to talk too loud to don't disturb the neighbors too much. So I hope you can all hear me, and welcome to my presentation on elemental impurities as we see them in USP and as we approach them in USP. But you will see in a second that it is very much in accordance, in congruent to what is also mentioned in Q3D, for example. So let me share my screen with you. So you should be able to see my screen now, or at least so I hope.
Yes.
So, so what we will talk about this morning or your afternoon there, or no, also your morning still. I think it's 11:20 or so in Indonesia. So we will talk about impurities in drug substances and drug products, elemental impurities. Here we are looking at, and look how USP is approaching that. And when we see the potential sources of drug impurities during development, we see here on the left side of the production chain, so to say, we see here basically all the organic impurities that come up and that are dealt with according to the Q3A, Q3B guidance of ICH, and this is basically where my expertise is lying, on the organic impurities more. And when we look on the other side, on the left, on the right side here of the figure, then we see reagents, ligands, solvents, catalysts, and this is basically the site where we have Q3C on the solvents being important and relevant, and then also the Q3D guidance from ICH on the elemental impurities. These can come basically, the elemental impurities that I will be talking about this morning will be coming from reagents, from the ligands and catalysts basically, but we also have them, of course, to a certain extent, maybe in excipients. Elemental impurities in excipients can also come up. I mentioned Q3A, Q3B, Q3C, and Q3D. I will not go into detail on these ones. I would just like to mention that for all these type of impurities, we have also chapters in the United States Pharmacopoeia, in the USP, and they are basically in accordance with ICH. There is not much of a divergence in those chapters, and I will talk to you today about the chapters that we have for testing on the elemental impurities only. So that's what we are going through in a second. Before I come to that, quickly about how the documentary standards in the USP are set up. We have at the basis of the USP, to understand it from the general terms, we have the general notices and the requirements there. And there we have in the general notices, just a second. In the general notices, the basic assumptions, the definitions given, and default conditions, how to interpret, how to apply the USPNF. And it's very important that this is read as well. And we will have a quick look into those general notices when it comes to impurities and then elemental impurities in a second. And then comes on top of that comes the general chapters, which normally contain common analytical procedures. And there we have chapters that are numbered less than 1,000. And these are the chapters that we call mandatory chapters, but they are only mandatory and applicable then when they are either referenced in the general notices. And we will see in a second that for the elemental impurities, it is referenced in the general notices, for example, but it can also be referenced in a monograph or in another general chapter that is numbered below 1000 here. Those chapters that are numbered above 1000 are informative, but not necessarily required. And then we have also the general chapters that are numbered greater than 2000, and those are mandatory chapters or applicable chapters for dietary supplements. We will look in that quickly when we look into the dietary supplement elemental impurities option. And then on the top, but not so important for us today, more important on the organic side, we have then particular specifications for each official article in the monographs. But elemental impurities, as I said, they are dealt with directly from the general notices on. That's also important to know. The same is true for residue solvents in the USP. You will not find in the monographs for residue solvents in all pharmaceutical article monographs, you will not find their tests on residue solvents, for example. But that does not mean, of course, that you don't need to test for residue solvents. That is also discussed in the general notices and is made then there mandatory for all pharmaceutical articles. And it's more or less the same for the elemental impurities. There we also have the section in the general notices directly saying that all drug product articles for which monographs are there in the USP need to be checked on elemental impurities according to the chapters that we have 232 and 233 in which we will go in a second here. So that's basically how the documentary standards are working in USP. And let's have now a look more into the inorganic impurities here in bit more detail. So today, for my talk, we are just interested in the inorganic impurities that you see here. We have some specific tests still on chloride and sulfate, for example, some non-specific tests where we check by residue and ignition, for example, on a sum parameter of basically that is then again all metals that are forming in the end non-volatile sulfates. They are then seen as a sum parameter in that non-specific text. But the most important chapters that we have now are the ones that we are calling elemental impurities limits and elemental impurities procedures here. You see here on the residue solvents, per the general notices 5620, it is applying to all monographs. That is also true not for all monographs, but just for those in the drug products for inorganic impurities. I will talk to that about that in a second when we go to the general notices, which is directly now the case overall. So the general notices, as I mentioned, very important to read and understand the whole setup of USP. And there is the section 560, which is dealing specifically with impurities and foreign substances here. And also to mention that the section 560 is currently under revision. There was this year in March, the PF 52 was published in March, and there we have a new suggestion, a new draft on the general notices, and also under 560 there are a few changes there, not so much on the elemental impurities, but on other parts, and we will come to the second to the elemental impurities in a second here. So when we look on elemental impurities alone here, that we are dealing with in 232 and 233 in the chapters, then we look here on environmental contaminants, and that is also including environmental contaminants, is also maybe including the elemental impurities, the metal that can come from the machineries, from the vessels, for example, in which we are producing our APIs, our drug products, if they are made of metals, for example. We also heard about the ligands before here and reagents, but basically the most important source of elemental impurities, especially the heavy metals there, and the most problematic ones are of course the catalysts there. And all those elemental impurities, they can occur naturally. We have some excipients, for example, that are mined naturally, and there we have, of course, then also elemental impurities in there. But they can also be like catalysts, for example, have been added intentionally. And also, of course, interaction, as I mentioned, with processing equipment and the container closure system. Also with that, we can, of course, introduce elemental impurities into the final drug product here. There's the possibility, of course, and it should be a risk-based control strategy to be set up, and that is normally appropriate to ensure compliance in the chapters themselves. You need to be compliant with the limits in the general chapter 232 at all times during the shelf life of the drug product, and we will come in a second to those limits and what options you have available there. So I mentioned already that the section, the general notices are currently under revision, and also for the elemental impurities, there was a slight change in the general notices. You see here that we aim to have this section here, this language here official in August 2025, where we are talking about elemental impurities in the USP drug products, but more specifically then not about impurities, but we call them then in the dietary supplements elemental contaminants. And here you see then the section where our general notices are saying you have to work in every official drug product. That means every drug product for which USP has a monograph in there, that every official drug product there need to be controlled on elemental impurities according to the principles of the limits, which are in 232. And in the dietary supplements, it is the chapter over 2022-32, for example. And methods, to which we are coming in a second as well, are then discussed in our chapter 233, but we will come to that a little bit later. So that are the three chapters that we have here, the general chapters. As I already mentioned, we have the limits mentioned here. Those limits are in compliance. Well, let's say they are the same limits as we have them in ICH Q3D guidance here. And we have then the chapters, the chapter 232, 233 with the two analytical procedures in there, ICP OES and ICP MS possibilities here. And we will discuss that in a second as well. And then the chapter 22-32 about the dietary supplement dosage forms. So before we come to the chapters a little bit more specifically, you might know that there is also that old chapter 231 that were giving us some parameter. You were kind of precipitating all the heavy metals as sulfates. You had then a kind of gray, slightly opaque solutions that you compared to a standard solution here on the and not the opacity, but how clear the solution is there. But that is, of course, a sum parameter. It's a very old wet chemistry test, of course. You know, wet chemistry is sometimes specific, sometimes not. But sometimes in laboratories nowadays, we always look to go to instrumental analytical work there. And that test that we had there, the sum parameter, it was more than 100 years ago that we have set up that chapter and introduced it. And it can fail, of course, when you look at mercury, for example, at toxicologically relevant levels, that can fail then there. The test and provide, not provide, but pose them some threats to the patients, of course. So that chapter is still in the USP, but it is made inactive. You can see it on the top, the chapter 231. It's still in there, but it is taken out of work. So, but for reference, it's still mentioned there. Have a look there. Also have a look, please, on the frequently asked questions about elemental impurities that you find here under the link as shown. Let's come now to the chapter on the limits for elemental impurities, chapter 232. The chapter is not applying to all of those that you see here on the slide. Yeah, basically because there are some special products where also at times metals need to be added at a higher concentration than the limits in 232 would allow, of course. Nonetheless, you can always use the limits in 232 there as a kind of guidance, and in case that you going above that, you can discuss with your regulatory authorities, of course, on these specific ones, and discuss each limit here separately. The chapter 232 is also talking about speciation quickly. It's not referring too much on that, but we mean with that there is could be different oxidation states in there. Organic complex, so we have arsenic and mercury, for example, that can also be present in organic, also other metals, of course, in organic complexes, and that combination we term that one speciation. And for arsenic and mercury, we need to be particularly concerned here. You will see that especially when it comes to the dietary supplement in the chapter 2232, excuse me, that there's mercury given as an element, but then also element alone, standalone here, but also then in organic complex, we have separate limits for that there. So here we have the limits, or it's just apart from the table number 1 in the chapter 232, where we have the limits for the elemental impurities based on the permitted daily exposure. And you see that here, and that was probably also explained when Dr. Loft went through Q3D this morning, that there are the different limits for oral consumption, parenteral consumption, and inhalation, and also newly. Now we have cutaneous products in there with a permitted daily exposure, and then also a limit for cutaneous and transcutaneous concentration limits here for sensitizers, where we are not going from the permitted daily exposure, but where it is thought that the sensitizers work on a concentration basis, and there we are giving a concentration rather than the permitted daily exposure. And you see here for cobalt and for nickel, we have here limits for these sensitizers, as well as a new limit for nickel on the inhalation permitted daily exposure. That is the table 1 based on the permitted daily exposure. And when you look then into the 232, you have 3 options for demonstrating compliance against the limits that are given in table 1. So you can do the drug product analysis option. That is that you take a typical dosage unit and check this one on the contaminants, on the traces of the elemental impurities. You scale then that result up to the maximum daily dose here for that typical dosage unit, and compare it then to the daily dose permitted daily exposure here. So, for example, if you have 4 tablets that you need to take as the maximum daily dose, then you test 1 tablet, and it should not be more than that. The tablet should not have more in there than a quarter of the impurities here from elemental impurities from the oral permitted daily exposure. You have also the possibility to use the summation option. In that one, you are not testing on a typical dosage unit, but you are testing all the components separately of the drug product. And you know, of course, how many of each component is coming into the drug product later, and you are scaling this then up to the final result, and compare that final result then to the daily dose permitted daily exposure here. And then the impurities should, of course, not be more than that one. It could be that in individual monographs there is another limit given then for a certain elemental impurity, but if not, then it should be as it is in the table 1 here. Another possibility, and that is also one that is often used, is when the drug product with a daily dose, and daily dose here of not more than 10 grams, we mean really the weight of the dosage form in the end. So the 4 tablets, for example, that might weigh together 10 grams or not more than 10 grams here. And for that, there's that individual component option, so that we have a limit shown, concentration limit shown in the table 3. And when all the excipients and the drug substances from that formulation are meeting the concentration limits shown in table 3, and provided the drug product is consumed not more than 10 grams here, then these components can be used in any proportion together. So that is what we provide here as a table 3, where you see here we are talking not about the permitted daily exposure, but again about concentrations. And if your excipients and the drug substance are fulfilling for an oral dosage form, for example, all these concentrations here, and you are not going above that, and the final dosage form is not having higher dose than higher weight than 10 grams, then you can use all the excipients, drug substances in any proportion together, and you will still be, of course, below the permitted daily exposure here, or not more than the daily exposure. And again, also, we have added here the new limit for nickel and on inhalation products, and then cutaneous concentrations, and the cutaneous and transcutaneous concentration limits for the for the sensitizers, which are, of course, unchanged to the values that you see in the table 1, of course. So that's basically about the limits in 232. Here we have then 233, which is then in combination with the limits of 232, is then replacing now what we had in that old bad chemistry chapter 231, which I mentioned as well, and which is nowadays not used anymore and inactive there. And I don't want to go too much into the procedures here. The most important, let's have a look how the chapter is set up. So we have a slight introduction in there, and then we are talking about the compendial procedures from the procedure side themselves, the detection technique, and how sample is prepared. Very often you need to do a digestion of the sample, closed vessel digestion we are mentioning in the chapter. And then we are also talking about the necessary reagents and the necessary grades of these reagents before we are discussing in a bit more detail than the procedure 1 and the procedure 2, and then how you should work with it. Of course, ICP OES, ICP MS is not present in all laboratories. Very often for smaller companies, medium-sized companies, that is a technique that might be too high in investment here. But of course, there are normally in these cases then contract laboratories that can provide such services. If you want to keep that in-house, for example, but don't want to use ICP OES, ICP MS, there's also the possibility to use alternative procedures there. And you can then validate those procedures and see if they are, of course, also as powerful and can have the necessary performance in order to be okay for the small traces that we need to find here. So limit of quantitation, limit of detection is of course very important there when you validate those procedures. And we also have a glossary in this chapter 233, and I would like to go quickly to that part of the glossary and just an excerpt from that one glossary here. In the chapter, we are talking about a matched matrix. And when we are saying matched matrix there, it's explained in the glossary that we mean with that solutions that are having the same solvent composition as the sample solution. So, for example, sometimes you need to to use aqua regina, for example, then also the standard solution should have the same concentration of aqua regina in there as that you needed the one for dissolving your sample there. And when you need to use mercury stabilizers, then also in both the standard solution and the sample solution, of course, should then be in there that mercury stabilizers at the same concentrations. We are also not working with fixed concentrations for all the limits in the chapter. That would make it very not difficult to write, but would expand it quite much to an extent that might not be necessary here. We are working here with the concentration called J, which you can also see explained in the glossary there, that we mean with that the concentration in weight per weight percentages of the elements of interest at the target limit. And then it is appropriately diluted to the working range of the instrument. So, for example, when we look on lead and arsenic, we have here the target limits of these elements in a 10 gram per day unit size or dosage form. We have the target limits here of 0.5 micrograms per gram and 1.5 micrograms per gram. But this is, of course, a solution that is too high for the linear dynamic range of the ICP-MS, for example. So for the ICP-MS, it is known to extend here over several magnitudes here, from 0.01 nanograms per milliliter to 100 nanograms per milliliter. So we are talking about 5 magnitudes, if I calculate that correctly. So to come into the range here from the target limits to the range, to work in that range, dynamic range, we need to use a dilution factor of at least 100 here, and that would mean then that that J would be 5 nanograms and 50 nanograms per milliliter for lead and arsenic respectively. So that's how we are dealing there with the concentrations. At the beginning, it might be a little bit not strange, but feeling awkward to accept. But once you get used to that approach, it's a very, very easy one. So that's basically already also with the chapter 233 that I wanted to mention here. And quickly about the elemental contaminants in the dietary supplements. So that's the chapter 2232, above 2000. These are applicable chapters always for dietary supplements. And it's basically set up in the same way as we have seen the limits are working for the elemental impurities in drug products in the chapter 232 here. So we have these elements that need to be checked. And you see here we have mercury as total, and then also methyl mercury calculated as mercury here, and permitted daily exposures here as well. And these methyl mercury methyl mercury is, of course, much more toxic than mercury as an element alone in an inorganic state. Therefore, we have, of course, here the lower permitted daily exposure for methyl mercury. We are saying in the chapter that when you can show that the limit of mercury is not more than 2 grams, not more than 2 micrograms per day, then it is not necessary to check also on the organic mercury in there. So when you are below overall, below 2 grams, not more than 2 micrograms per day, why am I saying 2 grams? 2 micrograms per day, then you don't need to check on organic because, of course, you are then below, by nature, of course, below that limit there as well. And the same as we have seen for 232, the same options to demonstrate compliance for the dietary supplements are here possible as well. So you have the analysis option to analyze the dietary supplement, one dosage unit maybe of the dietary supplement, and then calculate it up to the typical servicing size here. and then compare that to the PDE. You can also go with individual component option there. There's a table which you can use as well, so that you can see when these concentrations in each of the component is not reached, then you can also mix for a maximum daily intake of not more than 10 grams a day. Can also mix those components, means the working principle and all the excipients they're together in every combination that you want. And you can also use, of course, the summation option. So each component to be analyzed directly, and then you scale it up and see what is in the end present in each in each typical serving size, scale that up to the maximum daily intake and take it from there. So exactly the same option that you have in 232, you can also use for dietary supplements. And yeah, that's it basically about the chapters. We are not referencing there any reference standards in the chapters, but we have now reference standards available that you can use there, or you can also use certified reference materials from other sources as well. But you can work, of course, also with the quality reference standards from USP here, where we have class 1 and 2A mixture, class 2B mixture, and a class 3 mixture here too. And we have education courses on elemental impurities. The one, the broad ones that is giving a much more detailed overview than what I did just now is available on demand there as a recorded course. And we also have a complementary course here on the impact of elemental impurities in biologics development, manufacturing, and risk management there. So that one is complementary, and you can access it free of charge. And with that, I am at the at the end of my presentation. I'm not sure if Jenny showed this slide this morning to you as well, but please think about to work as a volunteer as a USP expert here. Under the QR code that you can see here, you can apply for becoming a USP expert volunteer in the next year. Our next cycle is starting. We are always working in 5-year cycles. And in June next year, we are setting up the new there's the convention coming together, and are selecting, electing the chairs, the committee chairs, and the committee chairs will then look into the amount, into the pool of all the volunteers that applied here to become an expert volunteer. They will look into the pool and of potential candidates, available candidates, and will set then up their committees there from that pool. So you are very, very welcome, and please look there. Tell us about your expertise, knowledge, so that our chairs can then later select you as part of the committee. And with that, I say thank you for listening to me this morning. And let's see if there are any questions in the chat or elsewhere. And for later questions, you also can, of course, contact Ela or myself here. Thank you very much.
Okay. Thank you, Dr. Christian, for that insightful presentation. So we moving to Q&A session. Please, ladies and gentlemen, you can raise hand to for question. Okay, maybe I will read the question in slide.com. Okay, Bu Nina, yeah. Yeah, please, Bu Nina. Okay. Okay, maybe
Bu,
yes, Bu
boleh
Nina.
saya pakai bahasa Indonesia?
Ya, silakan Bu Nina.
Ya, saya mau bertanya terkait dengan flavor dan coloring yang tidak terdapat di monografi farmakope manapun, ya. Apabila kita ambil persyaratan dari produsen bahan bakunya, di mana di dalam COA produsennya itu persyaratan elemental impurities itu cukup banyak, Prof. Jadi ada AS, ada
Sesuai dengan yang
himbal.
dibikin itu apa?
kadmium lebih dari 1. Nah, apakah seluruh elemental impurities tersebut harus diuji atau seperti apa? Mohon penjelasannya. Terima kasih.
Oh, Dr. Christian,
Saya sudah cantumkan di
ya,
chatbox juga sih.
ya, Dr. Christian, Ibu Nina have type a question in chat. May I ask Prof related to the elemental impurities in excipient? If the excipient doesn't exist in the monograph pharmacopoeia, e.g. flavor, coloring, we usually use COA from producing the excipient. Yes, if there are a lot of elemental impurities. Dr. Christian? Oh.
Excuse me. Yeah, I was I was coughing before and was putting myself on mute. Sorry. It depends on how well you you know the producer of this excipient, for example. So I would think that in the beginning, when there's no experience with the producer, that you should not trust then directly the certificate, but also should see when there is nothing written in the monographs, and we have no monographs for such an excipient, that you look then also, of course, and don't trust the certificate too much, but try to check it as well with the methods maybe from 233 there, and see if the certificate is giving the right amounts. That might mean that you need, of course, to give that task to a control laboratory. But I wouldn't trust the excipient producer, the supplier there directly fully on the certificate of analysis. There might be some issues with regulators when you see that. We at USP, we are not saying anything to that. That is more a task that you need to, or a thing that you need to discuss with the regulators. But that is a normal approach that you test a little bit more when you don't know the source too much, the supplier so much. But when there is building trust, then later, of course, you can also do a kind of risk assessment and skip the one or the other testing, definitely. Yeah.
Thank you, Dr. Christian. Next, I will read the question via slide.com. Please, my team, you saw the question. Okay. Should we check the elemental impurities for raw material for parenteral? Is it enough just create justification based on COA from raw material manufacturer?
Yeah, that is basically the same answer I would give there. That is more or less what we have there at the first question. And for parenteral, it is maybe even more important to also not rely just on the COA, especially when it is coming from a material manufacturer that you don't have too many experience with. Yeah.
Okay, next question. If there is no elemental impurities parameter in the monograph for parenteral product, should we still need to check it based on the COA review of the raw material?
I would say, again, that is more or less the same question. It's based on risk assessment, as I mentioned before. And you don't necessarily need also you don't necessarily need to have a monograph in here for those materials. Of course, they need to be generally regarded as safe, those materials, but they don't need to have a monograph. You just take them and test them as well, or you test them in the product and see then where the limits are then in the end. But basically, just relying on the certificate is not what you should do. Okay, there's the next question.
Okay.
How about the solvent that we use during manufacturing process of product? Is it need to check? That's a question today. That's one on residue solvents, where we didn't discuss too much here today. But you can work there also on in a way of risk assessment. Get some knowledge in the beginning. Again, that is not something that USP is normally commenting on. That is again something that needs to be discussed with the regulator. USP, the pharmacopoeia, is not a regulator here, but is giving tests that you can apply and limits there. And again, here I would say it is of course important that you have some knowledge about your product, how the residues, how the solvent in the end might still be there as a residue solvent. And with each experience that you get over your process control, over your process and the control of it, the more experience you get, you will see that you can adopt your testing here and then just check periodically based on the trend. But directly in the beginning, that is probably not something that you can do. But yeah, risk assessment is of course possible also in residue solvent testing.
This question from chat, the requirement of elemental impurities is only for the dosage form, oral, parenteral, and inhalation.
Yeah.
Does it mean there is the requirement for the other dosage, for such as ophthalmic solution, including the raw material for production?
Yeah. No, ophthalmic solution is also necessary to test. It is basically now added and that is why we changed the guidance. When you saw what I showed on the table, there was the limit for oral dosage forms, for parenteral and inhalation, but there were also now cutaneous and transcutaneous limits and given there. And an ophthalmic solution is then considered being cutaneous, transcutaneous. So that's for the eye here, that is considered then as cutaneous, transcutaneous. So it needs to be checked as well.
Next question. Yes. When we choose drug product option.
Well, that is completely depending on you. For example, has to do a little bit with the first questions that we had. When you have at some point maybe quite some knowledge about your manufacturers, your suppliers of the excipient of the drug substance, for example, and you know they are all reliable and trustworthy, and you can kind of justify that, then you can take from all the CFAs maybe the values then and do just the summation option and don't need to do drug product analysis here anymore. It's also depending on who is doing the work for you of testing on the product. There might be laboratories that have already, for some of your products or for the components that you use, they might have already some values there that they have tested for somebody else. There might be databases available with some service providers, and whatever is available, you analyze then what is available, and then you choose your best option. There is no from USP, there is no kind of recommendation when the drug product should be analyzed, or all the single components there for the summation option, for example.
Okay, Dr. Christian. Via chat, there are questions from Bu Nina. For optometric oculenta, we must test it elemental test.
Yeah, that is. I think that's the same question as we had before in the Slido. Ophthalmic products need to be tested for elemental impurities as well. Yeah.
Okay.
They are cutaneous products to that regard. Yeah.
The next question from via Slido. In USB monograph, mention for sodium hydroxide test limit of natrium and potassium using AS and also mentioned Ethernet using ICP. Our equipment will give equivalent result.
I would guess so. I mean, we have it mentioned in the monograph. So both methods are of course working normally on alkali metals, so natrium, sodium, and potassium should be both detectable very well with AAS and then also ICP. So that should be possible, I would guess. Whatever method is not described in the monograph, you need to validate that. So the methods that are mentioned in monographs, you only need to verify, which means also you need to take some aspects from the validation and test them in your laboratory with the method that you have transferred then into your laboratory, of course. But when you are using completely alternate methods, even when they are mentioned in the monograph there that ICP is possible, you would need to validate, of course, develop there and then also validate. Yeah.
Okay, thank you. Next question. Okay, from Pak Gede via Zoom. From what I know, I think it's a long time the elemental impurities USPGC from being proposed to becoming official. What are the obstacles?
Yeah, very good question. Thank you very much for that one. There was a long time where ICH, for example, was having guideline Q3A, Q3B, and Q3C. And there was a lot, for a long time, no progress that much on the Q3D, on the elemental impurities. And USP started there already way before the first drafts of ICH Q3D were available to work on that, and were suggesting limits on their own from other sources, and also methods were discussed there. And that was then brought into drafts all the time. And then so at USP, there was the elemental impurity methods and limits developed and suggested, and then at some point ICH came up with Q3D as well. The limits were then slightly different there, and then USP needed, of course, to adopt those limits from ICH, so that we are not saying different things. So that was a long time of developing. It's not always like that, but the obstacles that were mentioned here, or that are mentioned here, are of course so that different guidelines need to be considered and need to be taken in consideration, so that at the end no guideline is opposing any or contradicting any other. So that's why it also might take some time when the chapters are becoming official. But for Q3D, for the elemental impurities chapters of USP, that was specifically so because it was then developed before Q3D, then came Q3D, and we needed to take the limits then from Q3D there as well. So that was one of the reasons why it took also so long.
Okay, Dr. Christian, the last question from via Zoom. How to distinguish between injectable and oral grades, considering that the USP monograph does not directly mention whether certain ingredient as injectable grade or not?
I'm not sure if I understand that question correctly, but I would say, of course, in the end, what your drug product should do. Is it injected, or is it taken by oral consumption? And based on that, you are using then, of course, the limits of the 232 chapter. I'm not sure if I understand the question correctly. If I'm not understanding it correctly, then please can the questioner mail again, and we can take it then from there.
Okay, yeah. Thank you, Dr. Christian Zain, for the excellent session. All questions have been your answer. Ladies and gentlemen, before we close the session, I would like to thank you for all your engaging question and for being such an attentive audience. Thank you very much for all of your wonderful speaker, Dr. Jenny, Dr. Karen, and Dr. Christian for sharing their expertise with us today. Thank you for having me as your moderator. And I apologize if there is something unpleasant, and I will hand the session back to the MC. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you for all speakers and moderator for the valuable presentation and discussion. Excellencies, distinguished participants, ladies and gentlemen, before arrive at the end of today's agenda, I respectfully invite Director for Pharmaceutical Production and Distribution to give summary and closing remarks. Please be invited, Ibu Dita Novianti Sugendi Argadirija, SSI APTMM.
Thank you very much for the opportunity. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and best wishes for us. Honorable guests, our speaker, Dr. Jenny, Dr. Karen, and Dr. Christian, ladies and gentlemen. On behalf of Ministry of Health, I extend my sincere gratitude to everyone who joined this virtual workshop on ethylene glycol and the ethylene glycol, EG-DEG, and elemental impurity testing. A collaborative event between the Indonesian Ministry of Health, USP, and TGA Australia. The event was a success with all presentation delivered very smoothly. I believe we all have benefit from insightful discussion and had valuable opportunities to exchange experience in handling EG-DEG and elemental impurity testing. This workshop highlights the critical importance of ensuring the safety, quality, and efficacy of medication, which is shared responsibility between the government, stakeholders, and the community. The continuous enhancement of drug testing capabilities must keep pace with advancement in science and technology. International collaboration through shared experience and training is essential to strengthening the capacity of regulators and the pharmaceutical industry to meet standards, including the application of modern technology. I would like to express my special thanks to our distinguished keynote speaker from USP and TGA, who generously shared their knowledge, and to everyone who contribute their best efforts to make this event success. We hope that this collaborative activity can continue in the future with various topics and themes that meets our need. I hope this workshop inspires the pharmaceutical industry to implement EG-DEG and implemental impurity tests to produce the medicines that meet safety, quality, and efficacy requirement. Thank you once again for your valuable contribution, and I look forward to seeing you at a future event. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon. Thank you very much.
Thank you. Thank you very
Thank
much.
you, Director, for the closing remarks. Excellencies, distinguished participants, and ladies and gentlemen, this is the end of workshop on ethylene glycol, the ethylene glycol, and elemental impurities testing in collaboration between Indonesia Ministry of Health, the United States Pharmacopoeia, and Australia Therapeutic Goods Administration. I am sure we all gain a lot of knowledge and understanding from today's event. On the screen, we will show you a QR code. You can find the presentation through this QR code. Okay, lastly, I am Okti Aliviana on behalf of the organizing committee, expanding our appreciation to all of you for participating in this workshop here through the Zoom meeting. We hope you have a great day, and once again, thank you. Assalamualaikum warahmatullahi wabarakatuh. Waalaikumsalam.
Thank you very much. Terima
Thank
kasih
you
Bapak
very
Ibu.
much. Terima kasih Bu Direktur. Terima
Thanks a
kasih
lot.
Bapak Ibu.
Thanks. Bye-bye.
Thank you Dr. Christian, Dr. Karen. Akhirnya selesai juga.